So, welcome to this session of the Undergraduate Research Conference. For this panel, we will pause in between presenters to allow for questions. Please raise your hand and wait to be called on. When a question is asked, please repeat the question and to ensure that everyone on the Zoom and everyone present here is able to hear properly. So, each participant will have 15 minutes to present their work with about 10 minutes for questions in between. Presenters, I will give you a sign that you have reached the 10-minute mark by holding up my hands. This is the STEM and Health Sciences panel. Our first panelist is Ethan Wilkins, and his presentation is titled Comprehensive Detection of Endogenous CFTR Expression in Epithelial Cell Lines, Biochemical and Molecular Approaches.
Yeah, so like she said, my name is Ethan Wilkins. I'm a junior biology major, and I work in the lab of Dr. Henna. And what I'm going to be presenting on is what I did my summer scholars research last summer on, and it's comprehensive detection of endogenous CFTR expression in epithelial cell lines, biochemical and molecular approaches, a big mouthful. So at first, we're going to go over some terms and definitions. So what we do in our lab is we work on the delta F508, which is the most common mutation that causes cystic fibrosis. So what is cystic fibrosis? So it is a genetic autosomal disease that is caused by a mutation in the CFTR gene, and then that causes some sort of change or misfolding or lack of expression in the CFTR protein that causes the symptoms that are related to cystic fibrosis. So that can include, most commonly, so the thick mucus in the respiratory tract that can cause infertility in the reproductive tract, that can cause problems in the digestive system due to the pancreas not working properly. So it's a whole slew of problems, and there's over 1,000 different mutations that have been classified so far for cystic fibrosis. What we work on specifically is delta F508, which is commonly prescribed to about 70% of cases so far. So CFTR stands for cystic fibrosis transmembrane conductance regulator. I will refer to it as CFTR. It is a plasma membrane protein that is found on the apical side of epithelial cells that acts as a chloride ion channel. And this affects the electrochemical balance and gradient of the cell. And what happens is it allows for that transportation of chloride ions. And in response to the movement of the ions, we have the response of the movement of the water, which creates the difficulty and the symptoms that are related to cystic fibrosis. So I'm going to be referring to endogenous and exogenous cell lines. So endogenous cell lines are just natural expression, just the same as my cells and your cells. So there's been nothing genetically modified. It's just natural expression of the cell line specifically. So exogenous cell lines is what happens is you can purchase anything on the internet. There are generic just cell lines that have low amounts of your desired protein. And what can happen is there's protocols and procedures to go through where you can genetically modify, transfect that cell line with your desired expression of whatever. And then you can go through those tests that makes it easier to work with. So our cell lines, the CFB 4110 minus, that is that generic cell line that you can purchase. Years ago, it was genetically modified, and that's what we're using now through many, many generations later. So this is the non-transfected human cystic fibrosis bronchial epithelial cell line, and this will act as our negative control because it has so little amount of that residual CFTR, which is what we want. So our CFB wild type, so all CFB wild type is this negative control that has been genetically modified to express our wild type or normally occurring CFTR expression. So it is also a human cystic fibrosis bronchial epithelial cell line that was transfected with the wild type CFTR gene. And in this project, it will act as our positive control because it has so much upregulation due to the modification. Our last two cell lines will be our CIFBAC and CAPN1. These are endogenous, so naturally occurring. So CIFBAC is a human pancreatic duct cell line expressing the delta F508 CFTR mutation. And then CAPN1 is also a human pancreatic duct cell line expressing wild type CFTR. So both of these were extracted from pancreatic tumors in patients. It's just CIFBAC the patient also had cystic fibrosis, while in CAPN1, the person had the pancreatic tumor that was removed, but they did not have cystic fibrosis. So that's the difference in the two. So what is CFTR? What's kind of its function? I've talked about a little bit of that. So like I said, it's a plasma membrane protein that functions as a chloride ion channel, and mutations causes the protein to be misfolded, such as delta F508, impairing its function and transportation to the plasma membrane. So as I mentioned earlier, so what happens is this normal function has been known to part of the superfamily of the ABC transporters, which is commonly known in many cell lines. And its function is to allow the transportation of chloride ions and other substances, such as like bicarbonate and other things of that nature. And it helps regulate that gradient within the cells. And when that gradient is broken in cystic fibrosis for the respiratory tract specifically, we have this accumulation of very thick mucus due to the lack of water that is allowed to be transported to the mucus and allow for the cells to move it out and do its normal function of the respiratory tract. And what it causes is this thick mucus creates a breeding ground for bacteria. So most common procedures of treating cystic fibrosis patients is to treat them with antibiotics, because they usually come in with some sort of bacterial infection of the lungs and other things like that. So that's kind of the reasoning behind it. And like I said, it has a bicarbonate transport function that inhibits function of the pancreas and other things like that. So it can cause a whole slew of problems in all ductal organs where epithelial cells are found. So this misfolding of the protein, specifically delta F508, which is what we work on. So we have three bands, two are more distinct than others. So band A is just newly synthesized protein. So how proteins are made. So you have DNA, which is transcribed in the nucleolus of the nucleus, and then that goes through post-transcription modifications to be removed from the nucleus, and then it goes through translation by ribosomes, either in the cytoplasm or on the rough endoplasmic reticulum, and then it's transported through post-translational modifications to the plasma membrane or its respective area. So what happens is band A is that newly synthesized. It's just transforming from RNA to protein. And then band B is also immature CFTR, is where it's going through some modification, but due to a misfolding, whether it be the mutation or whatever, it gets caught in the kind of delivery system of the cell of the Golgi apparatus and doesn't make it to the cell, the plasma membrane, for it to do its function. So, and that is where we see a large band in cystic fibrosis patients. And then band C is a mature, fully glycosylated, fully modified, and fully transported CFTR that has made it to the plasma membrane, able to do its function. So what we see is on people that do not suffer from cystic fibrosis is that we see a large C band and a very little B band, while in people that serve with or suffer from cystic fibrosis, it's opposite. You see a large B band, so immature CFTR that does not do its function, and a very little amount of C band. So that's how we're able to tell the difference in these cell lines. So as I mentioned, this mutation causes a misfolding of the protein for this specific mutation, and it causes it to be caught in the Golgi apparatus and not allow it to make it to the plasma membrane to do its function. So my first technique that I will be discussing today is Western blotting. So Western blotting is a very common technique found in protein labs. I run a lot of them. Is a biochemical technique to detect a specific protein within a mixture of lysate, and it has RNA, protein, DNA, just a whole mixture of just biomolecules. And we have to be able to detect specific protein within that mixture. So what we do is we can run a experiment called gel electrophoresis. So it's made out of polyacrylamide gel that is used to separate these proteins based on size. Then the proteins are then transferred from the gel to a solid surface, such as a nitrocellulose membrane. And then I separate that membrane out and then expose it to different antibodies that are specific to our desired protein. And then we go through a series of steps to be able to detect that specific antibody under UV light so that we can actually see the difference expression. So here, this is comparing exogenous and endogenous cell lines. So in lane 1 and 2, we have our wild type exogenous cell line and our mutated version. So what we see is a strong C band in our wild type, which is what we expect, fully functioning CFTR. And in our mutated version, we see a strong B band, which is immature, not able to do its function is what we expect. But interestingly, if you look at our endogenous cell line, same. So we have wild type and mutated version, both, however, they're endogenous. We have not even a comparable reference between the two. But in these whales, we had to add 10 times as much protein to get not even comparable results. So in this one, we have 500 micrograms, while in this one is 50 micrograms. So not even a comparable. And so we're having to use more lysate, which uses more material. We have to use more antibodies, which is very costly to have these antibodies synthesized. So what we notice is that Western blotting is not a very effective way of analyzing endogenous cell lines for future research. Next up, I'm going to talk about RT-PCR, which stands for reverse transcription polymerase chain reaction. So this allows us to, so we lyse the cells and we are able to extract their RNA. And then we take that RNA and using reverse transcription, we're able to turn that RNA back into DNA using an enzyme called reverse transcriptase. And then the PCR part is what you do. It goes, you put certain enzymes in there and goes through a thermocycler, and you're able to take it from RNA to DNA and then amplify that DNA, which makes it much easier to work with. And this is a very useful technique for other protocols and procedures that I want to talk about, such as DNA gel electrophoresis. So very similar to Western blotting. So you go through gel electrophoresis for proteins. You can do it the same for DNA. And what you use for this one, you instead of polyacrylamide, you use agarose, and it still separates it using separates the DNA by like fragment of the DNA, the size of it, and you put a current to it, and it separates it. And kind of hard to see here, but I'm analyzing again negative control, my exogenous cell line, and then my two endogenous cell lines. And then on this side, this is my actin, which just serves my endogenous control, so that I'm adding the same amount to each, because it would be easy to say, well, this one has much more, you can kind of see it, well, it's easier to see, this one has much more CFTR expression, but I may have added 10 times as much compared to this one. Well, this kind of keeps the record of, hey, I've added the same amount to each well, everything's consistent. So using RT-PCR, I run a DNA gel, and I'm able to see this is our negative control, our CFB 410 minus, which is what we expect, a higher expression in our positive control. And then within our two endogenous cell lines, we're able to see a difference. We're capping one in lane 4, which is our wild type expressing endogenous cell line, having more CFTR expression than SIFBAC, which is what we've seen consistently throughout our analysis. And then we have this is a DNA ladder. So they commercially produce ladders that of specific lengths. So this one's 300, 200, and 100 base pairs, just so that we're able to know specifically where things are at in the gel, and we're able to have an idea of what's happening. So lastly, I'm going to talk about qPCR. So PCR still stands for polymerase chain reaction, but this one, Q, stands for quantitative PCR. So in that amplification of that DNA, of what I've talked about, we're actually able to quantify that by adding different probes and primers. And we're actually, instead of just having a before and after, we're able to see that amplification and then quantify that using relative quantification, CT means, and other things like that to actually create tables of the expression of each cell line. So there's two different methods. So first will be a dye base. This is when a qPCR first hit the scene. Dye base was the only way to do it. What it is you add a green fluorescent dye to each well, and then you measure that fluorescence intensity based on the amount of DNA that was amplified. However, there's a drawback to this, is because you can only add one specific dye, because all dyes were green at this time, to one well at a time for your desired. So you can either have, I'd have one well for CFTR and one well for actin, both having the same green dye, just different desired sequences tied to that dye, but I can't put them in the same well, and there's room for error in that. So what they came out, the new and improved, the probe-based qPCR. So I'm able to have different primers, similar, but I'm able to attach different probes that detect different fluorescence. So in the same well, in our machine specifically, I'm able to detect up to 4 different sequences of DNA within the same well that I'm adding everything into. So it's a much more accurate way of quantifying these RNA expressions within the cell lines and less room for error. So this is actually, it's an amplification plot. So I run a qPCR, it spits out something similar to this. And on my x-axis, I just have my cycles. So it runs 40 cycles of getting high and low and back and forth through the thermocycler. And on the y-axis, we have our amount of fluorescence. So what we expect is there's a certain threshold in here of when these lines pass that threshold, it tells me the CT value of that. So what we expect of a low CT value, so a low cycle number, for having more expression because it reaches that threshold even faster. So what we have is our 4 lines. So A is our wild type, which is our positive control. We expect a lot of expression. So this is kind of our 100%. Then B is our negative control, which is our just plain Jane generic cell line that we use for our negative control. And then within the 2 is our C and D values, which is again our endogenous cell lines. And what we're able to do is using relative quantification and CT values, we're able to compare these 2 cell lines versus the negative control with against each other. And we're able to see that CAPN1 had a 16-fold higher CFTR RNA expression compared to our negative control, while CIFBAC had a 6-fold increase compared to our negative control. And this is our same as the DNA gel. We have our endogenous controls sequence. But what this is able to do is instead of in the DNA, you kind of see a little difference here and there. There's some programs you use intensity of the band. Western blotting didn't work out, but using CFTR for a Western blotting, it takes me 4 days. You have so many expensive antibodies and all these things that I have to go through. I can run in one of these in about, if I have all my material, I can run in about an hour and a half, spit this out. I can create, as long as everything works like it's supposed to, create a CT value. And it's just much more efficient and cost-effective way of analyzing these different samples. So findings and future applications. So of course, as I stated, the RT-qPCR emerged as the most convenient method for detecting endogenous CFTR expression. Biochemical methods such as Western blotting, immunofluorescence, other things like that, requires large quantities of antibodies and proteins, which are expensive and time consuming. These detection methods hold promise in cystic fibrosis diagnosis because the use of these endogenous cell lines are what cystic fibrosis patients have. They have endogenous cells. So if we're able to find the best method to see how much CFTR expression they have, maybe we're able to detect the difference in severity. Maybe we're able to detect the difference in like, hey, what's a valid treatment if you have this mutation with this mutation? How much the different quantity of RNA expressed between those? So potential use in CF diagnosis and severity. And then CIFBAC holds significant potential as a parental cell line for transfection, such as the CFB41O- because it has such low amounts of residual CFTR expression, which is what we showed in our DNA gel and our qPCR. Any questions?
You spoke about the various different methodologies that you use to find expression, and you spoke to the one that has the most problems. Which one was the most fun for you? You look like you really enjoy doing it. I was just
well, it's like which one causes me the least amount of nightmares? Probably qPCR, because Western blotting, you go through the gel. So I have to make the gel, I have to load the samples, I have to take it out, I have to make sure the membrane's right, I have to cut it right, I have to expose it. There's just so many areas, and one thing goes wrong, and then it's just so finicky that now my future project that I'm working on with Dr. Hedda, I'm going to be doing a lot of Western blots because that's just kind of the way of measuring protein expression, because we're worried about protein rather than RNA. But I love running qPCR just because they're time efficient, and it's very there's high satisfaction because I'm able to spit out these amplification plots with this data and make like data points and assumptions within like the same day we're having these conversations, and then the next day or even the same day I'm able to do another experiment and just keep pumping out data. So it's really fun.
findings consistent with what others are finding in research, or is this something that's all relatively new in terms of what you're trying?
So this one, so not many people work with endogenous cell lines just because it's so hard. And most of the time you're trying to, like on another project, what we're working on is we're working with different compounds to see how they affect cystic fibrosis and help with maybe upregulation or transportation or all these different things. So many people work with exogenous cell lines just because they're easier to work with. So not a lot of people work with these cell lines. Like my professor told me that he believes that our lab was the first one to run a Western blot on endogenous cell line just because nobody does it. So but it's so important because when in real life, how are we able to best quantify this? If somebody comes in and say, hey, I'll try to give you somewhat of a result in 4 or 5 days, something goes wrong, we need more samples, whatever it may be, I can run a qPCR, like I said, in an hour and a half and spit some sort of data out as long as I've done my job correctly. So that's kind of the underlying behind it, is not many people work with endogenous cell lines. And because they are so hard to work with, so we were just trying to find the best way for people to potentially use them in future research. So, any other questions? Yes.
So when you did the summer scholar program, did you start out with an idea or had you already been doing research by the time you got into the program? And how has your project evolved since the summer scholar project till now? What have you been doing since?
So a lot of the projects that the people that come through Dr. Hedda's lab, so this project has been worked on a little bit by a previous student, and a project that I'm currently trying to transition to for my senior thesis was worked on by previous students. So I basically pick up where they left off and continue on. Now, I have questions for him all the time. I'll run a qPCR. I'm like, this doesn't make sense. This is not what it should be. And he's like, you're right. So we have to go through a kind of backtrack through another loophole of answering those questions. So I do have the freedom of having that discussion with him and be like, hey, can you explain this to me? And he's like, this is not how it should be. Or it's like, you've done everything you're supposed to do. Maybe we should backtrack and then move forward. So there's some freedom, but also you work with your professor. So he has projects that he is interested in for undergraduates, and then I can pick right up where everyone else left off and then continue on that research.
That was my other question. How did your project build upon other students? Because I have seen other students do some stuff that's similar to you.
Yeah.
So I see now how you're working together. And I like that continuum.
It's like if I graduate, whoever comes up else in his lab and that project is still going on, they'll pick up where I left off, and then it'll just continue on until everything we've checked all of our boxes, everything's ready for manuscript and publication, and then hopefully all of our names will be on it in the publication. Any other questions? Yes.
Maybe I missed it. What is DNA amplification?
So the way PCR works is you take a DNA and it goes through a series of different temperatures. So DNA will denature at a certain temperature. It's usually around like 90 degrees Celsius or something like that, depending on different factors. But what it does is there's enzymes, there's primers, there's materials in it. So once it reaches a certain threshold, the DNA, so it's double stranded, it'll come apart. And then what you do is you come back down in temperature. It's called the reannealing. So you add new stuff. So there's enzymes in there that can like replicate it. So then now instead of having one strand that breaks apart, now I have 2 solid strands of DNA, and that just exponentially grows 2 to 4, 4 to 16, and that just, so you have millions of different copies of DNA using this technique. And qPCR has been around since probably the 80s or 90s when it was first came onto the scene commercially. So it's been used all over the place. You had a question?
Oh, yeah, I was trying to ask your opinion, why is Western blotting not a preferred complementary test?
Well, it is for other, like for exogenous cell lines. It's we have programs that we can use to detect the intensity of bands, but just for endogenous cell lines, why it's not good for them is because there's so much room for error just in Western blots in general, no matter what cell line you're using. However, endogenous samples, like I showed, we had to use even more, 10 times as much sample to not even have comparable results, because those exogenous cell lines, they're genetically modified to show more of what you want, because you want to be able to see that difference in sample A and sample B with different treatments. about endogenous cell lines, it's just what is naturally occurring in the cell. There's no genetic modification. It's just like say you take a cell line of mine, it's probably cancerous, so to multiply. But say you take a cancerous cell line just from any human, put it in a petri dish, let it grow, it's not going to show this near the same expression as exogenous cell lines because of that, because they weren't genetically modified. So it's so much harder to see those subtle differences between cell lines from sample to sample. Say I use endogenous cell lines and I treat it with treatment A and treatment B. Well, it doesn't show, there's not much there anyway, so it's hard to tell that difference. Whether if I have exogenous cell lines with treatment A and B, it's much easier to see the difference between that because there's so much more there, so I'm able to see that difference. So that's why it's so much harder to use Western blotting in endogenous cell lines.
Could you see this at some point once you have identified the most cost-effective, most efficient, appropriate methodology for, I guess, expression and then treatment? Would this possibly trickle into human trials in like that?
So potentially, right now, like on my other project I'm working on with, we're using triazole compounds and treating different cell lines with that. So right now I'm running more of the protein side of it, but once I get done with the protein side or get all my materials together, I'll get on the RNA side. So I'll treat these same cell lines that I've been growing with different treatments, extract their RNA, use qPCR to see is there a difference in just general expression for these treatments. And then if we have data that supports different higher amounts of expression using Western blotting, we're able to see there's more transportation to the plasma membrane. We have tests to conduct, conductance of if they're doing their function properly, even though they're at the cell, the plasma membrane level. So then we get those samples from a lab in, I believe, in England. So we're running these, and then we see promise. We'll post that paper, and then potentially using them in future treatments. Because right now there's a treatment for, I only know of one for the Delta F508 mutation. What it does is it helps with that inhibited transportation from the Golgi apparatus to the plasma membrane. And it's a combo drug, and the other drug helps it kind of chaperone its unfolding to allow it to perform its function. So potentially using these compounds for treatments such as that. So, and triazole compounds are known for helping with chloride ion transport. So maybe help alleviate some of those problems, whether it be helps with function, transportation, expression, all these kind of things. So, any other questions? Thank you.
Our second set of presenters are Kiana Sanders, Anna McDowell, and Dr. Sarah McCrary, sorry. And their presentation is titled Natal and Neonatal Teeth in Mississippi Newborn.
I'm old school, so I'm gonna use the keyboard. All right, so hey, how are you guys? My name is Kiana Sanders. I worked on this research project last summer at UMC with Anna McDowell and Dr. Sarah McCrary, who was over the dentist over our project. All right, so first we will start off with what are neonatal and natal teeth. Neonatal teeth are teeth that are growth within the first 30 days after a baby is born. Natal teeth are teeth that are present in a baby's mouth at birth. This condition is relatively rare and occurs in 1 of 1,400 to 3,500 births. All right, so how the statistics on it. Using the Patient Cohort Explorer, which is our private patient database, we have identified at least 30 cases of neonatal and natal teeth in our patients. We expect this number to increase once approval is granted to access additional charts. The purpose of our research. The aim of our research is to determine common similarities between these infants in Mississippi. Factors we will review include gestational age, which is the age that the mother is during the pregnancy, maternal age, infant's weight at birth, the mother's diet, and infant's gender, male or female. The significance of our study. Natal and neonatal teeth pose a few issues. These teeth are often the mandibular mandible, scissor incisors, which are the bottom two teeth, which in return can create ulcers on the bottom of the tongue, leading to rigor feed disease. These teeth can cause issues for mothers who choose to breastfeed with troubles, latching, and painful feed. If the teeth are mobile, these may pose aspiration risk. And this right here is an example of what this rigor feed disease looks like. As you can see, the child has an ulcer underneath the tongue from the mandible incisors on the bottom gum. All right, so how will we be doing this? We will conduct this research by going through Patient Cohort Explorer, which is our private patient database, and accessing patient charts in EPIC using non-identifiable data to figure out the commonalities between these infants. We're aiming for patients born between May 1st, 2020 and May 1st, 2024. This will be a retrospective study, which is a study on past events and past data. In conclusion, in doing this research, we hope to better understand causes of natal and neonatal teeth in children in Mississippi. Additionally, this will allow us to better counsel and aid families with patients pronounced with this condition. This is our references, and thank you guys so much. Thank you to UMC for giving me the opportunity to do this last summer, and thank you to you guys for giving me the opportunity to present this information again. So. Questions or?
Have you heard of this before?
No, man, this is my first time hearing it on. That's why I was excited to be, being a part of it. So yeah, but we were just trying to get this off the ground when I started last summer.
Is there any statistics that are showing like Mississippi compared to other places that are having issues with evaluation of natal and neonatal teeth?
That's what we was working on. That was the honest broker was for, giving us information, having information from, I guess, other hospitals around Mississippi to let us know like how many other people have this disease. Like I said in my presentation, in our UMC, we have like 30 patients with this condition. Like I said, we were just trying to figure out now how common is it, like, in Mississippi.
We're just trying to, at this point, gather data to kind of see just how common it is.
Yes, ma'am.
So do you think this is something that could be caused by how old their parents are, or what sex the baby is, or
the parents' diet? Yeah, that's what we're thinking. Like that's why we said we're looking at things like the age, the parents' diet, maternal age, gestational age, stuff like that.
Or could it be heritable? Like if their parents had it, their grandparents had it.
True. Yeah.
How do you become interested in this topic?
I've always loved dentistry, period. I got my dental degree like I guess last fall. I got my dental assistant degree. So I've always had a thing for dentistry. And when I found out that they were doing a summer program at UNC, I couldn't wait to join. And yeah, they teamed me up with Dr. Sarah McCrary. We went through like something similar to this at the end of the summer and present our information. And yeah, I enjoyed every bit of it.
Is there a lot of data in other states?
That's what we're trying to figure out now. So when I was doing this last summer, we was getting this off the ground, like figuring out, who has what. Yes.
Are you trying to find out any needs with the prenatal thing? Like,
You saying it's like prenatal care? Is what you're saying? That's what we're trying to figure out now, like what is causing this situation, because it is painful and it can cause risk in many children if not treated. That's what we're trying to figure out now, like what's the all levels, so that way we can stay away from me, so we can all get down. Whenever me and her talked about it was a doctor overseas, like some kind of extension on the tongue or whatever. So in order to remove the ulcer, that was the one method I've known people to do, but we really didn't go into like the method of removing everything like. Could be. That's what we're thinking. We're thinking it's gonna be like the mother's diet, maybe, you know, stuff like that, kind of like any other mutation that kids have.
And I'm saying, we're more interested to see kind of like a comparison between a rural versus more of an urban area, those that have more access versus those who don't, and seeing if there is a prevalence in one versus the other. You know what I mean?
That's a good idea too. I definitely shoot her that idea.
I think it.
Yeah. Well, thank you guys so much for giving me the opportunity.
Our poster presentations will begin at 1 o'clock in the hallway. To this session's evaluators, please turn in your evaluations to the registration table as soon as you have completed it. For all attendees, if you haven't already, please remember to fill out the evaluation that is linked from the QR code in the conference program. Thank you for coming. Have a great day.